see on this phylogenetic tree that HCV is related to flaviviruses, including West Nile virus and dengue virus and pestiviruses over here. But because HCV occupies a unique branch on the flavivirus phylogenetic tree, it's known as Hepatitis C. Over here on the right are some of the other viruses thought to be associated with hepatitis viruses called GB viruses, A, B, and C. And GBV is actually hepatitis G virus, but it doesn't appear that it causes clinically relevant hepatitis. A few years later, they were able to delineate more aspects of the genome and genome function. And this basically demonstrates that the virus has an open reading frame of about 9,400 bases. At the 5' and 3' ends, there are these untranslated regions. The 5' untranslated region contains an internal ribosome entry site, which allows ribosomes to bind to the RNA and initiate translation of this large polyprotein. And in this zoom here, you can see on the left-hand side in yellow are the structural proteins, which form the three-dimensional structure of the virus, and they include the HCV core or nucleocapsid, the two envelope-like proteins E1 and E2, and P7 protein, which actually forms an ion channel that's important for replication. The remaining proteins, the NS proteins, are the non-structural proteins that have roles in virus replication. And so what happens is during translation, the ribosome makes this one large polyprotein, and then the individual proteins are released from the polyprotein by cellular and viral proteases. So some cellular proteins cleave the structural proteins, and the NS2 protease cleaves part of itself and then releases the NS3-4A protease, whose structure was solved in 1996. And so NS3-4A is responsible for cleaving these remaining non-structural proteins, which have roles in virus replication. So this is a cartoon of hepatitis C virus that, as Mike Yeldman may tell you, it's all over the internet and everyone uses it, but it was actually made by his daughter, Neil Gale, when they were living in Dallas when Mike was working with UP Southwestern as part of a science project. So it's actually a quite good picture, and you can see here that the viral nucleic acid is encapsulated by the capsid protein or the core protein. Around that is in yellow is the lipid envelope, and protruding from the lipid envelope are the envelope-like proteins E2 and E1. So this slide summarizes the virus life cycle, and like most viruses, or in fact all viruses, it has a typical virus life cycle. It involves binding of virus to cell surface receptors, internalization of the RNA, uncoding where the viral membrane is fused with the endosomal membrane to allow that genome to be exposed. The genome is then translated, the proteins are expressed on endoplasmic reticulum membranes, as shown in this zoom-in feature here. The proteins are expressed, the virus undergoes, the non-structural proteins convert the positive stranded RNA into negative stranded RNA that serves as a template for positive stranded RNA. And then virus is assembled at ER membranes, and as we'll discuss, at lipid membranes, and then the virus exits the cell. So this is a typical virus life cycle. And what we're going to do is we're going to focus on a few of these steps and talk about some recent progress in these areas. And first, we're going to talk about virus binding and entry. So over the years, there's been a few important advancements in the tools that have been to allow the study of HCV entry. And these are shown in this slide. On the left are the HCV pseudoparticles, which are basically gutted HIV virions studied with envelope proteins from other viruses. So these have been, pseudoparticles have been around a long time. And for HCV, basically they're just engineered to express the HCV envelope-like proteins. And they only measure virus entry, and they actually contain a reporter gene, a fluorescent gene in most cases, that delivers it to the infected cell. And because you don't require full replication, it allows you to study cells that do not support virus replication. And we'll talk about some data about using pseudoparticles in a minute. The other is tool to study HCV entry is the actual virus, this cell culture-derived virus called HCV-CC. So this is the authentic HCV virion, and it's generated from infectious JFH1 subtype 2A genome. It's capable of productively infecting cells in culture and animals, and it's detected by viral antigens or with incorporated reporter genes. So using these tools, investigators have found a number of cell surface proteins that mediate virus binding and entry. And we're going to go through this from left to right. So the first are glycosaminoglycans, which are linear polysaccharides that are present on proteins of all human cell surfaces. So these are these sort of random linear sugars that are stuck on the surface of proteins that stick out from the cell. And it's thought that these glycosaminoglycans provide a way for the virus basically to interact and stick to the cells. Then once the virus is on a cell, it can interact with several cell surface proteins, including this molecule, CD81, which was described as a HCV binder in 1998. And it's a tetraspanin superfamily member. It's expressed in all nucleated cells, and it's part of the B cell receptor complex. And then you can see here it has a number of transmembrane domains, extracellular cytoplasmic domains, and some intracellular signaling domains. In addition, there is another protein that's been shown to bind the virus, and this is scavenger receptor B1. It's a high-density lipoprotein receptor, and it can bind other ligands, and it's expressed in hepatocytes and various other human tissues. And furthermore, there's the protein claudin-1 in green that forms the backbone of tight junction strands in epithelial tissues. And interestingly, its highest expression is in hepatocytes. And I should point out here that if you silence any of these three proteins, CD81, scavenger receptor B1, or claudin-1, by silencing I mean if you block expression of these proteins, you can make the cell resistant to HCV entry. And one of the earliest putative receptors for hepatitis C virus was described a decade ago by Vince and Yellow and colleagues, and this is the low-density lipoprotein receptor. And evidence for the role of LDL receptor in virus entry is that viral RNA accumulation it correlates, either increases or decreases in parallel with LDL mRNA expression and LDL entry. So I wanted to tell you of this sort of this developing story about the role of tight junctions in HCV entry. And I wanted to show you a couple pieces of data with this protein called occludin-1, which is the newest HCV entry factor, and discuss the strategy that they use to identify occludin-1 and being involved in virus entry. So what they did is they had a cDNA library from these HUH7.5 cells. So HUH7.5 cells are human hepatoma cells that support HCV infection. So they made a cDNA library, put it in a retroviral vector, and they packaged it into pseudoparticles with different virus, VSV, the glycoprotein G. So they used the VSV-G pseudoparticles to introduce this cDNA library into mouse NIH3P3 mouse cells that had CD81, scavenger receptor B1, and claudin-1. They'd already known that if you put these three proteins on mouse cells, that HCV was incapable of infecting them. So they wanted to identify an additional protein in HUH7 cells that would allow entry into mouse cells expressing these three other receptors. So what they did is then they challenged with the HCV pseudoparticles, including an antibiotic, and then they tested surviving clones for their susceptibility to green fluorescent protein labeled HCV pseudoparticles. So the idea here is that if these cells get some cDNA that includes a receptor, it will allow it to be infectable with these HCV fluorescently labeled pseudoparticles. And using this strategy, they found that occludin-1 and CD81 determines HCV species tropism. And so what you can see here is this is measuring HCV pseudoparticle infectivity with this green fluorescent protein labeled pseudoparticle. And as a control here, you can see that HUH7.5 cells, just by themselves, allow really good pseudoparticle entry. And when you express claudin-1 on these cells mouse cells that express CD81, scavenger receptor B1, and claudin-1. In addition, when you express occludin-1, you can get pseudoparticle entry when CD81, scavenger receptor B1, claudin 1, and occludin 1 were expressed. Then they did similar studies in these Chinese hamster ovary cells that, again, if you don't have any of the receptors, they're not infectable. But if you express all 4 of these receptors, you can get good virus entry. And what was interesting is that this also looked at, they switched out human versus mouse for the 4 different receptors. And then you can see with the red arrows that the occludin 1 had to be human occludin 1 for virus entry to occur. So what these data indicate is summarized on this slide. And this is from a recent review by Thomas Piechman in Nature. And so what it is that the feeling is that tight junctions between these liver cells establish functionally different cell surface domains. So there's the luminal surface of the hepatocyte facing the blood vessel, and this tight junction allows separation of the hepatocyte with the bile structures, these canalicular surfaces. And over here on the right is sort of a cartoon of what's thought to occur, and that is that HCV is coated with lipoproteins. It binds to other things like glycosaminoglycans, possibly LDL receptor. It interacts initially with scavenger receptor B1 and CD81, and then it goes down, it's thought, down the side of the hepatocyte, where it encounters these proteins, occludin 1 and claudin 1, that are associated with these tight junctions. And I put they remind me of the guitar pick, so that's why I put the label on here. So it's thought that the virus comes across the cell, interacts with SRB1, CD81, goes down the side, encounters these tight junction proteins, and then allows virus entry. So now I want to talk about virus replication. And as I alluded to previously, HCV replication occurs on endoplasmic reticulum membranes. And in this circled part down here, you can see that many HCV proteins have membrane anchoring domains. So NS5A has an amino-terminal antipathic alpha helix that allows it to insert into membranes. NS5B has a C-terminal membrane binding domain. And you can see that NS3 is anchored in the membrane by its association with NS4. And so this is the non-structural proteins set up shop on endoplasmic reticulum membranes. And it turns out that not only are the non-structural proteins required for virus replication, but host cell proteins are also involved in this process. And I wanted to tell you this story about cyclophilin B, which came out a couple of years ago. So it turns out that cyclophilin B actually enhances HCV replication by directly interacting and affecting the activity of the HCV RNA polymerase. So cyclophilin proteins are these highly conserved peptidyl prolyl cis-trans isomerases that act, and they're famous for they're actually the receptors for the immunosuppressant drug cyclosporin A. But what normally these cyclophilins do is that this enzyme activity turns prolines from cis to trans isomers to facilitate protein folding. And so what this group did is they started working with various cyclosporin, these immunosuppressants, and various analogs of cyclosporin. And so you can see here, when they treated HCV-infected cells with cyclosporin, it had pronounced antiviral effect, and it correlated with inhibition of cyclophilin function. Sorry, my eyes bugging. Interestingly, there was this other protein that's a cyclosporin analog with reduced immunosuppressant properties, but it also could inhibit cyclophilin interaction, cyclophilin functions, but it was also a potent inhibitor of HCV infection. So they looked at HCV protein expression and RNA, and there was a really good correlation with the ability to inhibit cyclophilins and the ability to inhibit HCV replication. And when they looked at the association of the NS5B protein with cyclophilin B, and this is an immunofluorescence analysis, they found that NS5B was found in the cell in very close proximity to cyclophilin B. And they did some other studies to show that cyclophilin B interacted with NS5B, which is the viral polymerase. And when they measured RNA binding activity of NS5B to an RNA template. they found that the presence of cyclophilin B dose-dependently increased the ability of the viral RNA-dependent RNA polymerase to bind to RNA. It was completely separate. It was not dependent on cyclophilin A, which is a related protein but different, and that if you mutated the enzymatic activity of this peptoprolyl cis isomerase, you would lose the ability of cyclophilin B to increase binding of NS5A to its RNA target. So it's felt that cyclophilin B, because it can enhance protein folding, must allow NS5B to get on the RNA template. So this is a really good example of a virus usurping its cellular function to promote its own replication. Now where are we? We are going to talk about the last part of the virus life cycle, the assembly and maturation of virions. So this is a really recent paper, a very cool paper, showing that lipid droplets are the site of HCV assembly. And what this is an immunofluorescent detection of the HCV core protein in blue, the NS5A protein also in blue in a separate experiment, and lipid droplet-associated protein, ADRP, and staining of lipids with a fluorescent dye in green. So you can see here lipid droplets seem to be, if you look at the merged images, there's the lipid droplets in the middle, surrounded by the 4 protein and the ADRP protein. And this schematic over here shows, summarizes these fluorescent images. The lipid droplet is associated with the core, and around that is the NS5A protein. And it's been known for probably about 10 years that the HCV core protein interacts with lipid droplets and actually messes up lipid metabolism. But this was the first study to say that this interaction of core with lipid droplets was important for virus assembly. And the schematic of this is shown in here. You might ask me, well, you just told me that HCV RNA replication occurs on ER membranes, but then assembly occurs on lipid droplets. So how the heck do those two processes connect? And this is the model that the guys who published the study proposed. And you can see here, there's ER membranes here that has ER lumens, and they kind of interdigitate with lipid droplets. And so if you take this region here, which is this ER lumen with an ER membrane, you can see here that the viral non-structural proteins are replicating, they're inserted into the ER membrane, and they're replicating HCV RNA. So if you can, if this was a time-lapse thing, just imagine that this complex is moving around these membranes and it's traveling down here. So at some point it's thought that during replication, when the replicase encounters HCV core associated with a lipid droplet, that there is this transfer of this RNA onto core, which is also the nucleocapsid, and allows virus assembly at the lipid droplet. It's currently not known what are the signals that mediate this switch from this replicative mode into this assembly mode. But it is possible, at least, to get replication complexes near lipid droplets, the site of HCV replication. So these interactions are possible, at least geographically, within the cell. So in summary of the HCV life cycle, there are multiple proteins that mediate HCV entry, including CD81, scavenger receptor B1, claudin-1, and gluten-1, very low density lipoprotein receptor. The input HCV RNA is translated, polyprotein is formed, and individual viral proteins are released from the polyprotein by viral and cellular proteases. The HCV proteins associate with endoplasmic reticulum membranes, which is the site of HCV replication, and that the virion assembly occurs with lipid droplets. And for time's sake, I didn't have time to mention this, but it turns out that HCV also leaves the cell after it's assembled at lipid droplets by hitching a ride on the apolipoprotein B secretion pathway. So it's important to note that the HCV life cycle is intimately tied with lipid metabolism. So now I'd like to start talking about HCV variability. And as I mentioned, HCV, and as Chiron discovered 20 years ago, HCV is an RNA virus, and it's RNA-dependent RNA polymerase, or the NS5B protein. It lacks proofreading function. And mutations arise during replication are therefore not corrected. And this ability to generate mutations basically all the time during virus replication allows the virus to be grouped into genotypes, which are genetically divergent HCV isolates that can be grouped phylogenetically, and quasi-species, which are highly related yet genetically distinct viruses. And what I mean by that is summarized a little more succinctly on this slide. The term genotype is defined as heterogeneity among different viruses, and the nucleotide similarity is about 66 to 69%. When the viruses start to be similar, a higher degree of similarity, 77 to 80%, they become subtypes, which are closely related viruses within each genotype. And then within a genotype, there are quasi-species, which are this complex of genetic variants within individual viruses, and they're much more genetically related, about 91 to 99% similarity. And so this genetic heterogeneity and genotypes is depicted on this slide, and you can see that each genotype, genotype 1 over here, genotype 2, occupies a unique site on this phylogenetic tree. And you can see within each group, like Miriam Walter said last week, that there are type 1A, type 1B, and 1C. And so quasi-species, would be even more closely related within, say, this subtype. And just for comparison, I put up here the HIV phylogenetic tree. And what you'll notice is, what I want you to notice is that these scales are drawn to the same size. So this means that HIV is much less genetically divergent than hepatitis C. And it's probably the reason for that is that HIV has been in the human population for a much shorter time, and it's thought that the jump from primates into humans with HIV occurred much more recently. So HCV has been in the human population much longer than HIV, and that's why it's more genetically divergent. So quasi-species are kind of like siblings. They're highly related yet genetically distinct, and these are my two daughters. That's Alex and Maddie, and I guess they're jumping in front of the pyramids in Mexico. I guess they're kind of looking like a phylogenetic tree. And Alex is older, and her branches are a little longer than Maddie's. But anyway, I just put that up there. It's a good picture. Okay, so Miriam last week told you guys that Hep C, most people who get Hep C don't get rid of it. About 80% of people who get exposed to the virus become chronically infected. And so there's 179 people infected, and it's a big global health issue. So we obviously have been doing many things, well, not me personally, but there's been many efforts devoted to getting drugs to fight the infection. And as many of you know, interferon alpha-based therapy has been the standard of care for over 20 years in hepatitis C. And basically, interferon alpha is a recombinant protein that all our bodies make, and that when you inject it into patients, it induces an antiviral response in those cells. And Mike Gale's going to have a lecture about interferon and signaling that leads to interferon and innate antiviral defenses, so I'm not going to cover that. And pegylated interferon plus ribavirin combination is now the standard of care, and Dave Gretsch is going to talk more about therapies. But all I want to talk to you about is that therapy has improved over 2 decades, but about half of all patients are not cured of their infection. Now, that's not entirely true, because if you look at certain genotypes of HCV, like genotypes 2 and 3, with current standard of care, you can have an 80% cure rate. But if you look at all comers, it's about 50%. And the reasons for this therapy failure is that there's viral factors, the genotype and quasi-species impact the response to therapy, and that the virus employs strategies to counteract cellular antiviral defenses. And again, I'm not going to go into that. I think Mike's going to go into that in his lecture. There's a number of host factors that contribute to the success or failure of antiviral therapy. And there's drug factors. Interferon has very nasty side effects, flu-like symptoms, depression, effects on the blood system, so it makes it hard to tolerate for some patients. It's also very expensive. And then also some patients are a little skeptical of Western medicine and don't want it. But so there's many, many efforts ongoing, and there's lots of money being spent on drug development for HCV, which is a good thing. But this slide is just to remind you that if you're a company, drug development is not easy. That's probably why I work in the lab, because drug development is just really hard. And basically you can see that it takes about 12 years to go from preclinical testing all through the various phases of clinical trials and FDA approval. And you start, preclinical testing a few years working in the lab and animals, and you look at safety and biological activity. You do phase 1 and safety and dosage, and then you get to phase 3 where you have to do thousands of patients and verify effectiveness. And then after all this, you still have to get FDA approval all the way along in that final approval. And so this is pretty sobering that 1 for every 1,000 drugs makes it into humans. And of those ones that make it into humans, only 5 receive FDA approval. So I actually did this on April 21st before I realized it was HCV's birthday. But these are, as of about a few weeks ago, the number of drugs that are in development against HCV. So by far and away, the biggest class of drugs are the drugs that are targeting the virus life cycle. The most popular one is the NS348 protease, because if you block this enzyme, you can block virus replication, because the activity of this enzyme is absolutely required for virus replication. There are a number of drugs targeting the NS5B, the RNA polymerase. There's 2 targeting NS5A, and there are some hitting other steps of the virus life cycle that we talked about, including virus entry. There are some more general drugs that are against cellular targets. There's some that are blocking cyclophilins. There's miRNAs and some other proteins. There's immunomodulators, including stimulators and inhibitors of immune responses. There's TLR9 agonists, and then there's drugs going against the inflammation that Hep C causes and the inflammatory response to Hep C, as well as disease aspects. And then there's some interferons. So there's about 50 drugs in development, and what's interesting is about half of the studies, not these ones, but another 50% have been canceled. So this is a tough business. And so I just wanted to talk a little bit about, so we talked about the virus life cycle, and because there's a dependence on cellular proteins for virus replication, there's a good argument that you could go after cell-based inhibitors. And as I mentioned with the cyclophilin B story, there's a couple of drugs in clinical trials to block cyclophilin B. And these are these analogs of cyclosporin that don't have the immunosuppressive properties. And there's also, it turns out that there's an antibiotic, a broad-spectrum antibiotic, nitazoxanide, that's actually been shown to have anti-HCV activity both in vitro and in vivo. So these are examples of drugs that attack cellular targets and can prevent virus replication. And with that, with the last couple minutes, I'd like to tell you a little, an alternative approach, possibly, to treating people or making people a little healthier. And this is complementary and alternative medicine. It's been used for centuries by billions of people. It comes in various forms in terms of including traditional Chinese medicine, complementary medicine in Japan, and herbal supplements in this country. And many people take these things because they're thought to improve liver function, what we call hepatoprotection, which can involve antiviral, immunomodulatory, anti-inflammatory, and antioxidant activities. And one could argue because these are all botanical extracts and because they contain a mix of botanical chemicals, there's a potential to hit both viral and cellular targets with one botanical extract. And one of the things that we're working on in the lab is this compound called silymarin. And this is from this pretty plant here. It's actually a noxious weed. But the seeds of silymarin, when you crush it and make an extract, the seeds of milk thistle, when you crush it and make an extract, it's called silymarin. And this is a complex of these compounds called flavonolignans and a flavonoid that comprise the bulk of milk thistle extract. And it prevents, if you give silymarin to various animal models, it can block liver disease in various forms in various systems. And probably because of that, and also by sort of anecdotal evidence that it's widely used by HCV patients as a hepatoprotectant. And despite some controversy on its efficacy, the clinical studies indicate that silymarin is very well tolerated and safe. And so here's what I'm thinking when we talk about hepatoprotection, in that these compounds, or many compounds, not just botanical products, that have anti-inflammatory, antiviral, antioxidant, and immunomodulatory activity. so we have some really good chemical chemistry collaborators who can take the seeds and get an extract, and they can purify the individual components in silymarin. So we can go from a gamish to pure compounds with the help of our collaborators. And the first thing that we did is we tested silymarin against HCV infection using the cell culture-derived virus. And we treated infected cells with 3 different preparations of silymarin. And this is a Western blot showing expression of NS3 and NS5A protein. And you can see that the silymarin blocked HCV protein expression. And as expected, the established antiviral interferon caused robust suppression of virus infection. And you can also buy this stuff commercially from the gold standard of chemicals in this country, U.S. Pharmacopoeia. And you can see that silymarin or milk thistle extract causes dose-dependent suppression of virus infection. And that not only involves inhibition of viral protein expression, but as detected by real-time RT-PCR, an inhibition of HCV RNA production. So here is just looking at the kinetics of infection of cells with JFH1 virus, the HCV-CC virus. And if you treat with interferon, you cause this robust suppression of viral RNA accumulation. And silymarin also blocks viral RNA accumulation, but its kinetics are considerably delayed with respect to interferon and not as pronounced as with interferon. And if you can do these really cool experiments. You can take infected soups from supernatants that contain virus from silymarin and control-treated cells, and you can put them on naive cells and measure how much virus is there. So you can see when you take soups from solvent-treated cells, you get and transfer them to naive cells. You can get this is a measure of how much virus is in the supernate. And you can see that when you take soups from silymarin-treated cells, that there's far less virus release. So viral protein, RNA, and assembly release is inhibited by silymarin. And just to sort of, we were pleased to see this study come out last December. And it gave us hope that our studies that we were doing in the laboratory weren't just a waste of time. And this actually is in people. So what this study showed by Peter Ferenczi in Vienna, what he did is he got previous interferon non-responders. So these patients had failed the standard of care therapy. And he gave them 2 components from silymarin called silibin. He gave it to them intravenously, a 4-hour daily infusion for 7 days in different doses of silibin. And you can see that silibin caused, just with the 7-day lead-in phase of daily dosing of silibin, it caused dose-dependent suppression of viral loads in HCV-infected patients. And then he took these patients and then continued them with silibin and then put them back on pegylated interferon plus ribavirin, and it caused further suppression of virus load. And what was interesting is that he was able to get some of these non-responders to become responders with this sort of silymarin lead-in phase. So this gave us some hope that silymarin, if you give it at least intravenously, has antiviral effects. And I should point out that antiviral activity of silymarin has never been shown when it's taken the normal way, which is when you take it forward. But that has to do probably with pharmacokinetics and dosing and things like that. So in summary, HCV is a positive sense RNA virus that causes chronic liver infection in the majority of people that it infects. Well, I really didn't tell you that. I think Miriam told you that. HCV is a global health problem. During its life cycle, the virus interacts with numerous cell surface proteins on liver cells, replicates in association with cellular membranes, and assembles lipid droplets. The virus mutates during replication, generating genotypes and quality species which influence response to therapy. Interferon is the main treatment option for patients with chronic hepatitis C. There are new treatments in development, some that target the virus and some that target cellular factors needed for replication. And I suggest that because patients self-prescribe botanical or complementary and alternative medicines, and a lot of HCV patients do this. There's been some studies doing questionnaires. So I believe it's imperative to understand how these compounds work and how they interact with more traditional Western-based treatments like interferon and the new small molecule inhibitors of viral enzymes that are in development. And so what's going to happen in the future? Well, I don't know. I think better drugs are on the horizon. There are 38 of the polymerase inhibitors in clinical trials. Hopefully, they have some unexpected side effects. They seem to be manageable, but I think the drugs are going to get better. One of the problems with interferon is the non-specific therapy that causes all these flu-like things. Maybe we'll get a vaccine. I'm not really sure. I'm not an immunologist. I didn't stay in Holiday Inn last night, so don't ask me about it. In terms of limitations, I think some of the newer drugs, they're really great, they're really powerful, and they work very well. Compliance may be an issue because some of them require very precise dosing and timing of dosing. And resistance to these small molecule inhibitors is going to be an issue, and that's probably why they're going to be used in combination with interferon plus ribavirin. And my personal belief is that funding for HCV is just primarily, Japan, Europe, and North America. And for some reason, despite the fact that HCV infects 5 times as many people worldwide than HIV, funding isn't really available on the global scene. And also, expensive medications won't work for all. And I really think that we need more affordable therapies that will reach patients in resource-constrained countries. And with that, I thank members of my lab, Jessica Wagner, Mike Austin, who's not here, and Jessica Brownell, and past members who aren't listed here, as well as NIH for funding. So I'll end there, and I'd be happy to take any questions.